hope you had a brief little break and get some snacks or maybe some lunch. We're going to continue with our next session of speakers, and these will be 10-minute science talks from our AWG community. And the first speaker in this session is June Samsoneth. Unfortunately, she is not able to be with us in person, but she did submit a recording, which I will play for everybody. So give me just a really quick moment to pull that up. And while I'm pulling this up, I just want to mention that for the Q&A session for these 10-minute talks, we're going to wait for all of the presenters to talk, and then we will do a 15-minute Q&A for all of the presenters. So when you enter your questions into Conference.io, if I can please ask you to put the presenter's name in front of your question so I know to whom to address the question. So with that, let me play June.
Hello, my name is June Samsoneth, and today I'm going to be talking about the effects of radiation on human, mice, and Arabidopsis in space. So there's a lot of challenges to spaceflight. Radiation is just one of those concerns. With radiation, we're concerned about health and safety of astronauts. We're worried about the increased risk of cancer, degenerative tissue diseases, and acute radiation syndrome, just to name a few things. In plants, we're concerned about morphological and anatomical changes, as well as DNA alterations and damage. My GitHub is at the bottom of the page, and there'll be more opportunities to get my contact info for any questions that you may have after this presentation. So for this study, I utilized previous studies to help identify larger trends, patterns, and correlations that we may not have with just looking at one single study. I tried to utilize the GeneLab multi-visualization software. I also used IDEP, OrthoVen3. There's a lot of different softwares to choose from now. And if you would like to replicate this or try this on your own, there's a lot of softwares to choose from. These are just what I used. So when I was conducting this, I was looking for a comprehensive understanding of the biological and environmental factors influencing molecular and biological outcomes of radiation in spaceflight. And understanding how different organisms change biologically and morphologically at a morphological and biological level. It's really pivotal in addressing the challenges associated with long duration missions, understanding how to grow crops on the moon, that sort of thing. And it's also really great at generating hypothesis and experimental design with a novel approach. So for my methods, I utilized 4 different studies with 3 different species, plant, human, and mice. I listed the studies that I used, and I used unnormalized counts for these. I attempted to use the new GeneLab multi-visualization software to analyze and cross-compare with my own analysis. The code that I used for my own analysis is on my GitHub page if you're interested in that. However, there were challenges to conducting this study. So originally I proposed 5 different studies, which included flies and worms, but there was issues with data availability. Not all of the data I wanted to use is RNA sequencing. And to keep things simple and precise, I only used RNA sequencing data for this. I also conducted a DNA methylation study as well, but that's on my GitHub page for a more in-depth look at the 2 different factors. But yeah, also with the GeneLab multi-visualization software, they use data that has the DSEC2 normalization, and not all the studies I was interested had that. And just for the sake of understanding how how easy it was to use tools and software, I just stuck with what was available. And so the pool of studies that I ended up having to choose from is quite small because of these limitations that I set myself to. Here's just a more in-depth chart of the studies that I use. This is also on my GitHub page. Okay, so for this, I'm going to be comparing a single study, this single human study, with the combined matrix. So for the combined matrix, I used human ortholog. Here, go to the next one. I used human ensemble and combined the human, mouse, and Arabidopsis ortholog into one data set and created a factors matrix based on the factors from the previous chart and analyzed it all. And so there is a big difference in terms of what's happening at the molecular and biological levels with this method of analysis. So here's, for reference, this will also be in my GitHub page. Just the single study, this is the KEGG for the single study, and this is the KEGG for the combined one. And the principal component analysis factors are here in the corner. And for the sake of time, because I only have 5 minutes left, I'm only going to be looking at PC1 for both of them and comparing them. So for the combo matrix, we have more alteration of biochemical and signaling pathways versus with the single study, the single human study, we have a more focused, we're more focused on protein synthesis, processing, and immune response. And so this will be up on my GitHub page if you're curious. There'll also be more comparisons with Arabidopsis as the ensemble that we're looking at, and then also mice. So this is the heat map that I used strain versus anything else because of the correlation with strain here as the PC1. I'm sorry if I'm rushing through this. It's so interesting, and there's so many things I could talk about with this. So I'm trying to get as much in as I can. And so I also went in and looked at the single genes that were had the most variation. And so with the single study here, we can see these genes are have the highest variance. And then I also have this one for the combo matrix, and they're quite a different set of genes. I have it here in a table. And with the combo matrix, there's a lot less genes that we're focused on than the human study. And it's very interesting because with the human single study, it's mostly ribosomal genes that have the highest variance, but with the combo matrix, it's more mitochondria and genes associated with phosphorylation and that sort of thing. And this whole radiation focused study that I've done is an offshoot kind of a larger combo matrix that I've been working on. And in that one, it doesn't necessarily focus on spaceflight. Or no, it focuses on spaceflight. I'm sorry, it doesn't necessarily focus on radiation. And these genes in the combo matrix here with the radiation there's a lot of correlation with these genes and a lot of similar genes / the same genes that we're seeing as the larger combo matrix one, which will also be up on my GitHub page. And so with the human study, we are more focused on ribosomal changes that occur in spaceflight and radiation. But with the combo matrix, it's more mitochondria, which is just very interesting. And so things to consider. Is this a holistic approach to analyzing radiation in a spaceflight environment? Yes and no. It pulls in more species, more studies, and it is a more holistic approach in some ways. When it comes to the GeneLab visualization portal, it shouldn't be a limiting factor. I limited myself to only RNA-seq studies, only studies that were available with the GeneLab visualization software right then, and it shouldn't be a limiting factor moving forward. And this is a very powerful approach to advancing space exploration in the future. And thank you so much for joining me today. I hope you enjoyed my presentation. And if you have any questions or comments, you can email me at my email here, or look at my GitHub page or contact me through there as well. Thank you for your time. Thank you.
All right. So our next speaker then will be Anu Shikarkar from the University of Saskatchewan. I apologize if I did not pronounce her last name correctly. But Anu, if you are on, may I please ask you to turn on your audio, video, and start sharing?
Yeah, can I just get a confirmation that you guys can see my screen?
Yes, we can.
Okay, perfect. All right. Okay, so hi. Oh, wait, sorry. Hold on. Okay, cool. Okay, so hi everyone. My name is Anu. I'm a final year undergrad student at the University of Saskatchewan in Canada, and today I'm going to be talking about systemic genome correlation loss in spaceflight. So to give you a quick overview of what we're going to talk about today, I'm just going to quickly mention the issues astronauts face and the problem we set out to solve, then talk about our methodology, go through our results and conclusions, and then I'd be glad to answer any questions as well after everyone's gone. So everyone here has pretty much a really good understanding of this already, but it's still worth briefly mentioning that spaceflight negatively affects pretty much every single system in astronauts, from muscles to pleural to cardiovascular system to even the immune system. Now, we know that these negative effects happen at both the organism and also at the cellular level, with spaceflight-induced mitochondrial dysfunction being a main cause for a lot of the issues at the cellular level. But our lit review for the study revealed to us that a large portion of current research focuses on either individual genes or gene families or individual organ systems. And there is research, but there's very little research looking at gene expression and co-expression patterns within the genome. So that's what we decided to focus on here. Now, because spaceflight is so expensive and difficult, one of the main issues in space medicine is actually the number of samples we can access in studies. So for that reason, we also attempted to develop a way to combine multiple studies from GeneLab for this research. So we started off accessing the raw data from these 5 studies that had both untreated control and untreated flight samples, and then internally normalized and pre-processed these datasets to basically combine all of them into master pre-flight and in-flight expression datasets. After that, we did a whole bunch of analyses on both the combined and individual datasets to verify our normalization and data combination methodology. So this is just a graphical overview of the methodology we used, as well as some info about the individual datasets we use. I'd be glad to answer any questions or anything you may have about this afterwards. So moving into our results, the first thing we looked at was principal component analysis, or PCA, to basically analyze our dataset normalization methodology. When we colored the data points based on the dataset or the original study that they're from, I think it's pretty clear to see here that pre-normalization, there's significant batch effects. You can see a lot of a grouping of similar colors together based primarily on mission profile. But post-normalization, we see a lot more uniform of a distribution, which indicates a good normalization. Now, we can also color these data points based on assay type and flight or ground profile. And again, we see significant grouping based on assay type pre-normalization, especially in PC2. But post-normalization, we see significantly more even and dispersed distribution of data points. And we also see something really interesting here, which is a grouping of flight samples together, which implies that the analysis we do further on is actually analyzing the ground flight difference and not necessarily any other flight effects or batch effects. Now, of course, when we have gene expression data, the first thing we do is differential expression and pathway analysis to look at, again, which genes and pathways change their expression. So here I've shown you the top 15 pathways, of which notably 12 are very, very heavily involved in mitochondrial energy processing, functions which, like I mentioned earlier, are already known to be hugely altered in spaceflight. So this is a good verification of both our normalization process and also our DG and pathway analysis. Now, before we continue to gene pair correlation, I'd like to take a second to actually explain gene pair correlation here. So if you plot gene A on an x-axis and gene B on the y-axis here, correlation between two genes basically tells you how likely you are to be able to predict the expression of gene A from gene B. Now, positive correlation basically means that when gene A goes up, gene B goes up. When gene A goes down, gene B goes down. So they move in the same direction, right? And you'd expect to see this in, for example, two inhibitors of a pathway or two promoters of a pathway. Negative correlation is when gene A goes up, gene B goes down, and when gene A goes down, gene B goes up. So they move in opposite direction. And zero correlation just means that you can't predict the expression of gene A from B. Now, the way these are represented is essentially by color. So hold on, I'm going to move forward here. Positive correlation is blue, as you can see here. Negative correlation is red, and zero correlation is white. And basically, the darker the color is, the stronger the correlation is. So this is what a correlation plot looks like. Essentially, here all you have is a list of genes plotted against itself. And so at each XY intersection of these genes, you draw the dot that corresponds to the correlation between those genes. So this is only about 30 genes, just so you guys can actually see the dots and for the example. But what we did first was do this correlation plot for the entire genome. So this is really interesting here. This is the ground, so pre-flight data set. For the combined samples. All of the data that I'm going to show you is for the combined samples, but again, we did do all the analysis for both the combined and the individual studies, and we got good results with both. We got similar results, so I'm going to show you the combined study. So again, this is the pre-flight dataset ground. This is 10,000 genes in one axis interacting with 10,000 genes here. So this is over 100 million interactions that we look at just in this image. But when you count replicates through the individual missions, data sets, and samples, there's over 10 billion data points represented in this image, which I think is always really cool to point out. Now, something to observe here is that pre-flight gene pair correlations are generally quite strong, right? Whether they're positive or negative, the average absolute correlation value here is quite high. Now, this makes perfect sense because the human body is a very, very organized system, and pathways work together to accomplish tasks. So you would expect to see strong organization in correlation as well. And you're going to have antagonistic pathways be very strongly negatively correlated and sympathetic pathways be, again, strongly positively correlated. But as soon as you bring in the spaceflight samples, so this is the in-flight sample, we see a huge change in gene pair correlation pretty much all across the genome, and we see a loss. A very significant loss, actually. So this implies that not only is gene expression significantly changing, but the genome itself is also starting to lose coordination as a whole, and possibly activating conflicting pathways at the same time, and potentially causing a lot of the cellular level issues that we see in astronauts, and also wasting energy in the body. Now, something else that's notable to point out here is clustering of these 10,000 genes to identify if correlation loss was happening in any specific pathways, which is what we did. So instead of doing a biological clustering based on pathways, what we did in the order of the genes, was a naive mathematical clustering. And we found that the correlation clusters we identified, for the most part, don't overlap with any known biological pathways, which means that genes that aren't classically known to be related to each other are somehow losing coordination in the exact same manner, which again implies spaceflight specific pathways, at least for this characteristic of gene expression. Now, speaking of pathways, we also replicated this correlation analysis on individual pathways. So here's an example now looking at neuronal genes, where we see a similar correlation loss. So of course we also did network analysis, and here you can see very, very dense networks, which kind of implies that both at the microRNA and the protein level, there's very likely to be a couple of controlling genes at the top that are then controlling the rest of the genome through this correlation loss and through this very dense network of interactions. So I know this is a lot of data and figures that I've just shown you. Just for take-home messages, I've condensed this into three main points that I think encapsulate most of this research. So firstly, that universal gene pair correlation loss is possibly a central characteristic of spaceflight in astronauts. We haven't seen anything like this in any samples on Earth, and this kind of large-scale genomic alteration, without necessarily changes in gene expression, is something that's completely unique to space. Secondly, it's possible that spaceflight induces unique biological pathways that are only activated in spaceflight, because genes clustered together in correlation clustering do not necessarily overlap with biological pathways. And lastly, that correlation clustering tends to overlap with known degenerative markers and health losses in our samples, and so presents a very exciting diagnostic monitoring and treatment opportunity for astronaut health. So before I wrap up really quickly, I wanted to take a quick second to acknowledge all of my collaborators and supervisors. Lauren and Sylvain, who are definitely in this call right now, have been absolutely incredible with their insight, advice, and direction for the project, as well as Dr. Lukong, Dr. Tagi Beglu, and Dr. Yang at the University of Saskatchewan, who were instrumental to the success of this project. This project also wouldn't be possible without the open data that GeneLab provides. So huge shout out to the GeneLab and OSER teams as well. We're in the process of publishing this paper right now, but the preprint is accessible through this QR code on screen. We also ended up actually having some fun and designing a patch for the experiment, which you can see right here. And I almost always have stickers to hand out at Space Bio meetings, so come find me if we're ever in the same room together, and you'll probably get some cool stickers as well. And with that, I'd like to say thank you to everyone here for your attention and to the symposium for giving me the chance to talk about some of my work. I hope you found it interesting, and I look forward to answering any questions. Thank you very much.
Great talk, Andrew. Thank you so much. So our next speaker, or we have 4 speakers for this next presentation. So we have Alessandro Paz Hernandez, Elaine Huang, Jennifer Wallner, and Adith George. If they're all from Purdue University. If you would not mind, please turning on your cameras and your audio.
Hello, can you guys hear me?
Yes, we can.
Hello.
Do we have Elaine and Jennifer and Adith on as well?
Yeah, can you hear me? I'm here, yeah.
Perfect.
So, hello everyone, we're Purdue DataMine, and we're with the Porterfield Lab, and we are going to present about our study about photo aspiration in space. I'm Alessandro Paz.
I'm Elaine Hong. I'm Jennifer Wallner.
I'm Adith George.
So we wanted to thank our collaborators, Purdue DataMine, Purdue Agriculture and Biological Engineering Department, Dr. Porterfield and Dr. Ward for making this possible, and Dr. Barker, Ruth, and Dr. Cruz for helping us dive deeper into this research with insights and direction. We want to thank our main team as well.
So when looking at our 3 data sets, being OSDs 217, 427, and 469, all of them focused around Arabidopsis thaliana. And the significance of Arabidopsis thaliana is that it's a C3 plant. C3 plants, compared to their counterpart C4 plants, are behind in evolution and aren't as adapted to an oxygen-rich environment. And as a result, in such an oxygen-rich environment, if there is a lack of flow around the leaf, rubisco tends to react more with O2 than CO2 and cause more photorespiration rather than photosynthesis. And this is significant in a spaceflight environment because with reduced microgravity, there's reduced gravity-driven convection or less flows. And as a result, O2 tends to form bubbles around leaves, which are just another indicator of the O2 concentration around a leaf. And the reason C3 plants are significant in the first place is due to them being significant in the agriculture space, with wheat, soybeans, all being examples of C3 plants that are crucial to humanity and society as a whole and the food production systems. But it's key to note that these studies are also important to be expanded towards addressing potential astronaut health concerns, as the spaceflight environment can be dangerous, and there are definitely factors that need to be considered when moving people beyond the boundaries we know currently.
Yeah, so like Audis mentioned before, in space, oxygen is stagnant around the plant, and this causes an increased oxygen, and this ultimately decreases the photosynthetic efficiency of a plant. Now, normally carbon dioxide would bind with the enzyme called rubisco to start the Calvin cycle, which is a crucial process for the plant that generates the energy and nutrients that the plant needs to thrive. But when there's excess oxygen, photorespiration basically competes with this necessary carbon fixation. And this photorespiration often happens in response to like environmental stresses, which leads to overall decreased productivity in C3 plants, especially. And while this photorespiration kind of serves as like a protective mechanism to prevent this damage caused by the oxygen, it can impact the growth and yield of the plant. And photorespiration is also a multi-organelle response, and this can affect multiple downstream pathways. And as you can see here, the Calvin cycle in gray only takes place in the chloroplast, but the photorespiration process involves the chloroplast, peroxisome, and mitochondria. And because of this, photorespiration consumes like excess ATP and produces excess byproducts in multiple steps of the pathway. And this energy expenditure ultimately affects the overall energy use of the plant, and it decreases growth and yield.
So for our experiment, we used 3 different data sets, those being OSD 217, OSD 427, and OSD 469. OSD 217 was a more simple experiment, just looking at molecular changes and how growth is affected in the Arabidopsis plant. OSD 427 focused on how terrestrial stress-associated epigenetic modulations impacted spaceflight adaptation, and this was done by comparing wild type and ELP ecotypes. And finally, OSD 469 examined changes of wild type and transgenic plants grown under both long day and short day photoperiod conditions. So for filtering, we use the typical p-value less than 0.05 for significance, and the log2 fold change helped us compare genes that were expressed at different extremes. In the table to the right, you can see the percentage of data that was filtered, and you can see that the columns are similar for each experiment, which indicates that the error is experiment dependent. And on the next slide, this shows just some of the factors and levels across the different experiments. 2 I want to point out is the first one, which is ecotype. 217 used WS ecotype, and 427 and 469 used wild type Columbia ecotype, among others, as you can see. And then another factor I wanted to point out was light cycle. So 217, 427 both used a 24-hour light cycle, where OSD 469 used both an 8-hour light condition and a 16-hour light condition.
So we went a little bit deeper and we mapped all these genes, the genes that were combined in all these different studies. And we mapped the subcellular location using Zuba-5. And we saw that, yes, plastic mitochondria and peroxisome were differentially expressed after that filtering. So then we dove deeper and we looked at the photorespiration pathway specifically, and we saw that about 80% of the genes in this pathway were actually differentially expressed. And so some insights and some comparisons that we want to do and show you are ecotype dependent and light dependent. So specifically in the ecotype dependent, OSD 217 and 427, run both by the same group in the University of Florida and have the same protocols and same conditions. And the only difference is the ecotype. However, there seems to be a really large difference, like almost 40 times difference in the genes that are differentially expressed. So we want to look into that further. Because of this, when we plotted into the reactome, actually 427 did not have any genes in the photorespiration pathway that were expressed. We don't know what happened, so we reached out and we're like communicating with the Florida team now. And in 217, we saw that rubisco upregulated 100 times. Mitochondria matrix proteins differentially expressed -4 to 100 times, and peroxisome matrix proteins 20 to 100 times upregulated. So now we're looking into the light cycle experiments. We wanted to compare 427, 469, long day and short day, which are 8, 16, and 24-hour conditions. They are the same ecotype, so this is why we chose them. And this is what we found. Basically, there's a lot of the genes that are shared between 8 and 16-hour durations, but again, 427 didn't have any genes differentially expressed for this pathway. So we went deeper and we actually plotted the subcellular location. So how did the differential expression play across the different organelles? And we actually found increased differential expression comparing the 8-hour to 16-hour light cycle, and also increase the same pattern, increased differential expression comparing 8-hour to 16-hour cycle in the mitochondria. And interestingly, we found the peroxisome only appeared in the 16-hour cycle. Then we looked at the reactome again, and we found, yeah, in the 8-hour cycle, mitochondrial matrix proteins upregulated 3 to 4 times. And compared to the 16-hour light cycle, the mitochondrial matrix proteins regulated 3 to 40 times, and peroxisomal matrix proteins appear to be regulated 2 to 4 times. So as any good bioinformatician would do, we plotted the heat maps. So in this heat map, you can see increased upregulation across light cycles. And this is on the gene level. So we went a little bit deeper and we wanted to look at the enzyme and the enzyme level response. And so we saw the exact same response and exclusively upregulation. So increasing upregulation across light cycle treatments. And as you can see here in the third column, you have Rubisco, and this is the enzyme that we wanted to check for specifically photorespiration, which seems to be really upregulated in the 16 and 24-hour cycle. So we went a little bit deeper and we also saw the organelle response, and we checked the mitochondria, peroxisome, and plastid. The three of them show the same pattern, increased upregulation, which kind of supports the hypothesis that we had of increasing, with increasing light periods, there would be increasing photosynthesis, which would lead to more oxygen around the leaves, in turn would lead to more photorespiration in the plant. However, we saw the cytosol had some downregulation, so we want to explore that further. So we saw this increased upregulation across light cycles. So we wanted to look at if adjacent pathways actually experience the same thing. And so we looked at amino acid synthesis pathways, so glycine, serine, threonine metabolism, and some preliminary results show that there's increased differential expression. And yeah, some conclusions is we found enzymatic organelle and gene responses from the respiration pathway to consistently increase in upregulation across light cycle treatments. We also found that the response to the light cycle treatments might be organelle dependent or precursed before Rubisco. And we want to look deeper at the biochemistry to adjacent pathways to see how, like how we show the amino acid responses, which can be further validated with single cell data and plant-psych type data and net space type. Thank you.
Awesome work, Alessandro, Elaine, Jennifer, and Adith. Thank you so much for your presentation. We'll take questions, Q&A after the session is over. So our next speaker in the session is going to be Andres Caicedo from Universidad San Francisco de Quito. So if you wouldn't mind turning on your audio and video, please.
Sure, here I am. Hello, Roberta. Thank you very much for the invitation and everybody. So I think I'm ready to share my screen. Perfect. Okay. So thank you very much to everyone for this amazing opportunity and invitation. I will present you and talk to you today about the forms of circulating extracellular mitochondria in space medicines. Are there victims, heroes, or sentinels? I'm also very proud to represent the USFQ Spacefront and an amazing group of scientists that have been working in this great project. Here are my conflict of interest statement, in which I perform scientific advisory for companies such as AWG, Drown Biomed, and also the Vice President-elect of the IECC team. So for many years, we've been amazed by the technology that's been developed, the great engines, how this technology is helping us to arrive to hopefully to the moon again, and also to Mars, and to reach, in matter of saying, the stars. And I think that this endeavor is the way for mankind to explore space, and also to have the capacity to bring all the exciting resources that will be available. But one key thing is to protect the health of the astronauts that are going to be part of the mission, and also to space tourists, to help them to come back to Earth in good health and also to live many years. One key thing that I believe that is super important is that the efforts of the AWG is helping us to understand better how astronauts are reacting to the stress, but also how we can solve problems from people here on Earth. So I think that is a spectacular mission of this group that will help us to live longer and better. So this seminal article was the base of some of the amazing research that we are performing, in which it was observed that mitochondrial dysfunction is a significant contributor to the health risk associated with spaceflight. So mitochondria is at the center of many of the deregulation that we could be observing in the muscle, the loss of bone mass, and many other factors that could be affecting the health of the astronauts during long-term space flight. So let's focus a little bit more on this amazing organelle to understand better their key role inside the cell, but also outside the cell. So first of all, the mitochondria is far beyond the powerhouse of the cell. It has a multifunctional role, and it coordinates the metabolic adaptation of one cell and also helps the cell to communicate with others in order to preserve our health, and also how to we respond to stress. So the mitochondria is supporting the cell in the physiological adaptations that they need to have in order to overcome stress. So the cell receives these inputs, modify their mitochondria, and have outputs that have an important role in maintaining our physiological health. It has been observed that we have, as different cells in our body, we have different types of mitochondria with a different metabolic programming. For example, we have stem cells that are highly glycolytic, and when these stem cells differentiate, they activate the mitochondria. Even they activate the ROS production in order to differentiate in other tissue types, for example, in skin and muscle and many others. And I wanted to bring your attention to this great article that was published in 2020, in which the team of Alan Thierry observed for the first time that there is mitochondrial competent in our blood, respiratory competent, and also intact, that is circulating in our bodies, and even when we are in good health. So this mitochondria is not inducing a pro-inflammatory response, but rather have key roles that are still under investigation and could have important effects in our health. So later, after the publishing of this publication, we developed a model in which we propose that there is a diversity and coexistence of these extracellular mitochondria in our circulation. So what could be the effects of mitochondria being transferred from one tissue to another, and if this transfer of mitochondria from one specific cell to another could be helping it to being able to respond better to cellular stress. And in a conversation with Dr. Jonathan Breston, the PI of these two great articles, he was able to mention it to me that the circulating mitochondria that is intact and is breathing have the capacity to being released to the circulation when, for example, adipocytes are unable to transfer these mitochondria to the macrophages. So when this happens is when there is obesity, and is also the exposition of fatty acids. So exposition of fatty acids, obesity, is hampering the transfer of the adipocyte mitochondria to the macrophages, and this mitochondria that is being released into circulation is going to the heart and also providing advantages to its function. So in this case, mitochondria, when it's outside the cell, in circulation, it is a hero, it seems. Then, by studying, for example, what is happening with when people are older adults or they are overweight, we observe that there is an increase in cell-free mitochondrial DNA. And however, in the analysis of the cell-free mitochondrial DNA could be related more to the presence of different types of mitochondria that are carrying this cell-free mitochondrial DNA, and that is associated with different responses, biological responses that need to be further understood. So this could be that these forms of extracellular mitochondria MTDA could be mistaken only for the cell-free mitochondrial DNA alone. And it was recently, together with Dr. Martin Picard, that we proposed different types of these envelopes of the cell-free mitochondrial DNA, or the cell-free or the naked cell-free mitochondrial DNA, and the possible implications that it could have in our health. Very interestingly, by the AWG group and a group of amazing scientists, such as part of the team of Dr. Christopher Mason, they observe in these liquid biopsies that these mitochondria is releasing, or it seems that there is the presence of the cell-free mitochondrial DNA when the astronauts are in flight. And then in another very interesting paper by the team of Dr. David Gokusen, that these same mitochondrial DNA is increased after the flight. So it seems that there is a certain dynamic that could be happening in the when the astronauts are going to space and then coming back to Earth. So it is super important to understand this better. One key thing that is super important is to try to develop better ways to understand and to decode the type of mitochondria that is present in the plasma of these astronauts, as it seems that sample manipulation may be hinder the identification of the different types of mitochondria and their roles, because when they are taken, they need to be preserved in cold, and this could affect the intact mitochondria, releasing their DNA and also affecting our hypothesis and understanding of what could be happening. So this part is super important in order to know the role if mitochondria are actually the victims of the cellular stress inducing a pro-inflammatory response, or actually, I mentioned before, heroes that are being released by the tissue, and that these mitochondria that is arriving to other tissue have an important role for maintaining our health. And this is some of the questions that we are trying to answer here at the AWG USFQ team, together with amazing scientists such as Dr. Christopher Mason, Ashim Beheshti, Ji-Won, Jan Keung, Elia Gino, and Andrea Camera, in which this part of the results were presented yesterday by Dr. Veronica Castañeda. And she showed that there is mRNA coming from mitochondrial genes in circulation in the JAXA mission. So this was very interesting to understand that different complexes, complex 1, complex 4 and 5, there is the presence of this RNA in circulation. So we are trying to understand this a little more and try to associate this with other studies, as I mentioned before, of Dr. David Gokusan, in which there is this RNA that is in circulation, but also there is this DNA that is in flight. So this seems to be part of a dynamic response that is happening in our bodies with the release of mitochondria. Later, after asking this, our team here at the USFQ infuse intravenously mesenchymal stem cell mitochondria in the mice, trying to understand a little more about their distribution. And we observed that these mitochondria are going to the brain, are going to the heart, and also going to the spleen, and have different effects. For example, in the spleen, it seems that 25 nanograms to 250 nanograms of these mitochondria that has been intravenously administered into the mice is increasing the red pulp size of the spleen, showing a pro-inflammatory response, and also inducing different patterns of expression of cytokines, such as those that are pro-inflammatory, but interestingly, those that are anti-inflammatory as well, such as IL-10. These results also go with the result of Dr. Gokusen, in which we observe that there's an increase of IL-6 and many others. However, we need to understand a little more, for example, why at the same time there is IL-10, for example. This could be related to different mitochondria that are in place. This is something that is worth to study. Interestingly, as I showed you, we detect by qPCR the presence of human mitochondria in the brain. We were able to also correlate its presence with mitochondria that were labeled with the mitotracker red in the mice brain, in which we observed that the presence of these red mitochondria was colocalizing with the vessels, and some of them were actually being transferred to the parenchyma of the brain. So this is very interesting for us because the presence of these mitochondria could affect, for example, the response of our brain and could actually have important implications in our health. So here, thank you very much, Lorca. It is super important that to understand that there is a possible dynamic that will possibly maybe hero mitochondria is increasing during the flight, but damaged mitochondria starts to appear and are eliminated after their return. So more studies are needed, and let's collaborate to further know what is happening with these extracellular mitochondria. And hopefully they have a hero mode of acting, which we could, for example, infuse astronauts with very good and healthy mitochondria in order to replace or help in the replacement of the others that are damaged. So here from the USFQ, I thank you very much to everyone, and also invite you to collaborate and to explore possibilities of initiatives in which we could develop cell science, maybe hospitality for space exploration, and the use of the different facilities that we have over there in Ecuador, in the rainforest, in Galapagos Islands, also in the Andes Mountains, to test different environments to further develop space science and space exploration. Thank you very much to the great team, because they are great. They're very motivated to move forward into what is space exploration. And please, if you need further information or let's start collaborating, please contact me to this mail. Thank you very much.
Thank you, Andres. That was a great talk. So we're running a little over. We were supposed to have a break now. So I think with only 4 minutes left, we should just continue, because I want to make sure we have plenty of time for Q&A, because there are a lot of questions coming in. So if everybody's okay with that, our next speaker is Matthias Nesset from McGill University. So Matthias, if you would not mind, please turning on your audio and video and sharing.
Sounds good. Share this. Okay. Now you can see my slides and everything?
Yes, we can, and we hear you perfectly.
Okay, perfect. Let me just put my pointer on, and then we can go. All right. Hello, everyone. I'm Matthias Nesset. I'm a PhD candidate at McGill University, and today I'll be talking to you about my project, the Cardiovascular Outcomes in Humans Exposed to Microgravity, a systematic review and meta-analysis. Today, I will be giving an update about some of the preliminary results we've gotten. So to start with some background, the original idea from this project stemmed from a previous meta-analysis my lab completed about bone changes in microgravity. We originally wanted to see if we could link these any of these bone changes to cardiovascular changes. However, when we went to do this, we noticed a lack of large-scale reviews preventing us from linking this cardiovascular data. Therefore, what we did is we went and we approached the ALSDA AWG in 2022 with the idea of completing a large systematic review and meta-analysis of all cardiovascular data in microgravity. And we asked them for their expert guidance on how to go about this and conduct this. So for a long story short, and what could and very much has been other presentations, this actually led to the development of the SOLSTICE program, which stands for the Space Open Life Science Team for International Collaborative Exploration. This is a citizen science program where we originally recruited 100 people from 28 different countries to help us undertake this massive project. We currently have about 25 active members who continue to contribute to the project when we have work to give them. Again, this part of the background is likely familiar to a good chunk of you, as I've presented stuff on this before, but I'll just briefly describe the steps that we took to develop the search. We consulted with a team of 20 AWG experts to develop relevant search terms, and had a team of multi-center team of 4 medical librarians run the search in Medline, Embase, CINHL, and they even included some articles that were published in GeneLab and had their data available. Here we have the resultant Prisma diagram from the work. As you can see, we began with a tremendous amount of articles at just under 20,000. After that, we began title and abstract screening, which is here, where actually a lot of the citizen science help was used. In short, we took this just under 20,000 articles that were screened in 6 weeks, with 75% of the work coming in just a small time period of 2 weeks. This led us to about 3,500, which were left for full text screening. And what we did is we divided these into relative subgroups, such as actual microgravity and humans, which I'll be assessing today. And this left us with just under 1,000 for that group that I just mentioned. From there, we had just over 200 included, and looking with cardiovascular terms, and we looked, right now we have about 72 looking at heart rate outcomes, which I'll talk more about later. So the tables on the screen here might be a little bit hard to read, but the idea behind them is that these are all the cardiovascular outcomes that we expected to be reported, which have been divided into 5 main groups as kind of denoted by the colors. Here we have the cardiac measurements, the pressure, vasculature, heart rate, and anything other that wasn't in there. So the figure on the right here shows the distribution of how these outcomes were reported in those 200 or so included articles. And what we did is we decided to start with the largest group here, which is the heart rate outcomes. Today, I'll be going over the preliminary data for 9 of the 72 heart rate articles, or about 12%. So just a little bit of an early insight into what we're doing. So I'll quickly just describe to you the methods behind the upcoming analysis for the absolute heart rate analysis we did. Here we have a random effects model to account for the heterogeneity between the studies, as it's typically more generalizable and more conservative than the fixed effect model. We also had 4 main groups / time points that were being evaluated. We had pre-flight, which included both seated and supine, as these values were not always similar. So we decided to separate them. We had in-flight values, and then we had post-flight values. Additionally, for this analysis, any of these groups that had several time points of measurement, say there were 3 different time points of heart rate for the in-flight, these values were, for this point, they were averaged, and then their statistical values were converted and maintained. So here we have the results in forest plots of the aggregated data heart rate outcomes during those 4 groups mentioned. One thing you might see like right away is in the pre-flight, in-flight, and post-flight group, we have this outlier. That's this is because one of the studies actually reported their outcomes at maximal physical exertion, therefore having significantly higher heart rate. While this is less useful in looking at just the heart rate on an absolute level, it will be useful for us for relative changes, which I'll discuss a little bit more later. But what I'll do here is I actually removed those outliers just so you can get a better idea of the absolute values here of how the heart rate is being shown. Here you can see that there might be, from the pre-flight values, you have 61 here on average, and then it moves into a 64 on the in-flight, and then post-flight it moves up to a 67. But by looking at the confidence intervals associated with these, you can see they generally overlap, especially like the pre-flight and the in-flight, meaning that there is the potential for no difference as well. That being said, we'll actually further explore these changes in relative analysis. So here, I just wanted to plot the changes all together so it'd be easier to see. So most studies reported, like I said, pre, in-flight, and post-flight values denoted by the numbers 1, 2, and 3 associated with the study. However, one reported pre and in-flight only, so no post, which was this one, the Carlson, and one reported pre and post and no in-flight, which is this one here. Again, you can see the outlier group here. But for the sake of the figure, I just wanted to show this to show how studies vary at different measurement points within each study. For example, if you look at CareMaker, you can see that it has a decreased heart rate, that in-flight has a decrease compared to pre-flight slightly, but a more increase in post-flight. This is also true in the seen in the Levine case 1996 here, and in the Otsuka 2016. But it seems that the Lee 2020 and the Marshall-Gopa 2019 have something opposite, where the in-flight seems to slightly increase, and then the post-flight is increased from the pre-flight. Now, here again, I just wanted to remove that maximal exercise group just to narrow down the range of the heart rate and just give a better visual so you can see the actual disparities between the study points. Now, I will provide a brief description about the methods we used for the relative change analysis. Overall, we kept a lot of the same previous assumptions, such as this random effects model. Otherwise, we used the standard mean difference model as the effect size, and we measured the differences occurring through 3 groups: the in-flight to the pre-flight group, the post-flight to the pre-flight, and the post-flight to the in-flight. I will note that due to the low number of seeded pre-flight measurements, that all the pre-flight measurements I'll be reporting from here on out are the supine group. Here we have the plots for relative change. As you can see, the in-flight to the pre-flight group does not have much difference, as the model is centered around the 0 value. However, if you look down to the post-flight to in-flight group just below, you can see that this model value starts to shift to the right, starting to indicate that there might be some kind of heart rate increase from post-flight compared to the in-flight values. Now, if you look at the post-flight to the pre-flight change, you can see that the model results have actually shifted well to the right and well beyond that 0 value, indicating a strong case that heart rate actually increases significantly after microgravity exposure when compared to the pre-flight baseline. Now, we currently do understand that our data is very limited, as we only have about the 12% of the articles that we want to look at. But we do find these preliminary results to be insightful, and we want to see how they might carry throughout further analysis. As I mentioned before, for a study like this on the table, where there are multiple time points within one of the groups, we averaged the values for these measurements. But we also want to look at how these might change over time, especially like during or after emission. So to do so, we are completing meta regression of the data. However, the results for this are not fully available yet, so I didn't have anything to present today. But I did want to mention that's definitely something we're looking into. Overall, the key takeaway from this preliminary data is that more work needs to be done to further analyze the relationship between the in-flight and the pre-flight heart rate. However, the post-flight heart rate seems to increase significantly in comparison to the pre-flight values. So after seeing these results, you might be asking kind of what are the next steps we're going for? Well, first is to continue this extraction and complete the rest of the data, obviously. But we also want to add in the heart rate variability analysis, which I'll talk a little bit more about the next slide. At the end of this all, we do plan on uploading our data and curating it into OSDR so that it can be reused and accessed by others. And finally, a smaller takeaway from this, but a big focus of just the work we've been doing in general, is that citizen science gave the participants a chance to contribute to the project, providing them with a unique and very helpful to actually us as researchers educational experience. As I mentioned, we are looking into the next steps of the project, but had a couple questions ourselves about the work. For example, in the case shown here in the orange box, how can we be sure that the data is not being reused in 2 different places? For example, the average days in space here is very similar between these 2 studies, and they have a very similar publication date with similar values. And considering they don't always report exactly where the data comes from, what missions and so on, we don't know. We're looking for insight on how we can kind of counter this or be more mindful of this. Additionally, as we move into the heart rate variability side, we're hoping to gain more insight from the experts within this group as to specifically which parameters to consider, because there's time domains, frequency domains, nonlinear measures, but we just want to get kind of the core of the idea. Overall, I'd like to acknowledge my PIs, Dr. Tella, Dr. Komarova, the many collaborators from the AWG and Solstice, the members of my lab, and the CSA and the NSERC for funding. Thank you.
Wonderful. Thank you, Matthias. Great presentation. Moving on to our next speaker. We have 3 more speakers in this category before we can get to Q&A. But our next speaker is Saswati Das from ABV IMS and Dr. RML Hospital. So Saswati, if you would not mind, please turning on your audio and video and sharing.
Hi, I hope you can hear me.
We can.
Could you enable me to share screen?
Marco, can you do that, please?
Yes,
Because
try now.
Go ahead and try now, Saswati.
Thank you. So, a very good evening and good morning to all of you. It's night in my part of the world. I'm talking from India. So this is a paper that we did in collaboration with AWG and the scientists from JSC, so Lab Medicine Space. And it's a paper discussing what is required for the real-time diagnostic laboratory medicine that is crucial to maintain astronaut health. At present, point-of-care testing and a few basic analyzers are available on the ISS. And there is a lifetime occupational survey of astronaut health that is conducted by the Flight Medicine Clinic at Johnson Space Center. However, there are a lot of challenges in spaceflight laboratory medicine, and there is a lot of requirements. So just introducing the team, a lot of you know Lo Afshin. He is a part of the NASA AWG Multiomics. He's chairing that. All of us know Fatihi is also one of the members of the Multiomics AWG. The other authors in this paper were Joe Wensek. He's an associate professor of pathology in Nashville. Dr. Kathleen McMonigal, she is a clinical pathologist at Johnson Space Center. Brian Cushion, a research immunologist at Johnson Space Center, and Dr. Kai Trudel from Ottawa is in physical medicine and rehabilitation, is a physician there. So they have all conducted experiments in space, and they've been working on how to develop robust space-enabled laboratory technology for future space missions. So what are the challenges in space? A lot of you conduct space experiments and would be knowing a lot of them. So serum collection and freezing at -80 degrees centigrade. The degradation of samples that happens, the hemolysis that happens, carbon monoxide exhalation has been used as a marker for calculating the hemolysis of the samples. Then how to ensure the return of the samples. And there are a lot of operational constraints as well in the payload design. Volume, weight, power, reagent stability, these are some of the main concerns. In microgravity environment, waste disposal is a big issue. So there is an international effort for miniaturized low-powered instruments, which is really essential to function on board. So as I was telling, blood collection supplies are very limited on ISS, and currently point-of-care testing device known as ISTET, which is manufactured by Abbott and has been used in microgravity, and ISS is present on the ISS for testing the astronauts. And there are certain basic analyzers, but those also there have been some degradation in the reagents that has been observed. There is a centrifuge machine, and there is a -80 degree freezer at the ISS. These capabilities are already there. So what are the challenges in space? So supply logistic is one of the biggest challenge, because they are the resupply is by uncrewed cargo vehicles, and the cost estimated is around $10,000 per pound, which is very, very high. There's a communication lag. There is sometimes inability to resupply. So these are some of the major concerns that have to be addressed in future. Then there is, although ISS is compared to a 5-bedroom apartment, but the actual space there is very, very limited, and that leads to the importance and rise of compact devices and compact payloads. And this hardware needs to be tested for chemical off-gassing and taste contaminants. It has to pass that, and the equipment functionality has to be there in microgravity. So fluid behavior needs to be studied and ensured that the instrument is functional. The reagents, the resupply reagents should be stable and should be tested in flight. There are many research challenges. There are many IRB issues about the de-identification of the data and the small number of astronauts. There is limited grants that are available and leading to a limited access in laboratory medicine research in space. So why lab medicine specialists are required in this, and what are the programs that are currently or in future will be available? So one is the commercially enabled rapid space science. It will facilitate dispatch of specialized scientists to low Earth orbit. It is going to be a program that is to be also in collaboration with NASA, and it will support research beyond astronaut capabilities. Orbital Research is also coming up with a similar program. Why are pathologists and laboratory medicine experts needed? Because they can assess the potential pathology risks. And what is required is that every mission might not have a lab med specialist. They will have to transfer the data on Earth, and the communication delay should be addressed, and it should be a real-time assessment of the data. So this specialist will be critical to the mission. And why is that important? Because some conditions need to be identified even before the flight, and the issues need to be resolved before then. So what is important is having a quality assurance and a quality control program. So how can that be achieved? That was a very elaborate discussion in the paper, the experts inside that we had in the paper. If any of you need an access to the paper, please do write back to me. I have my address written in each of the slides. So the pre-flight data and the post-flight data is very robust and it can be preserved. When you are testing in the ground labs, that is for the pre-flight data and the post-flight data, the ground lab needs to be accredited as per the criteria that we have. Mostly labs in US are CAP accredited. In other parts of the world, it is ISO 15189 accredited. Then long-term storage of the samples for future research, that should be available. Baseline testing of the return samples. Then there is something known as good laboratory practice. So that needs to be followed. This is a part of the safety regulations. There should be flight certified controls and standards that need to be tested on board. So as we understand, as of now, in flight, the quality control is not as robust as on Earth, but because of the non-availability of flight certified controls and standards with the instruments that we have. Then all the reagents need to be approved and should enable sample preservation. And cold storage facilities need to be available on board. So what is important is maintaining the data integrity. So what is needed for that? That is, there should be standardized processing, that is SOPs. We need to create SOPs. And one of the excellent example is the NASA Gene Labs SOPs that we have. And how can we validate our data? Some of the processes that have been tried is Dr. Trudel in his RBC's experiment, he tried all of this, uncoupling the experimental blood sample draws and synchronous sampling in conjunction with the earthly control. So these are some of the validation studies that can be done. Although on Earth, our validation studies are very, very robust. We check for the accuracy, the precision, the limit of the linearity range and the clinical reportable range. So maybe that robust validation might not be available on board, but then a mini verification has to be done as per the good laboratory practices. So how can lab technology undergo evolution for future mission? This was also a point of the discussion. So what we need is single device for multiple sample types. We want rapid analysis that can be also possible with mobile technology. Now, some of the continuous glucose monitors are astronaut wearables, and there are other astronaut wearables as well, like smart socks. So they can be coupled with the mobile technology, and those can store the data. Then reagent storage solution is a very, very important because there has been an observation of degradation of reagents, specifically with the analyzer, WBC analyzer that was used on ISS that was commonly observed. Then we need to have a quality control plan, which can be also AI enabled because we have those enabled in our laboratories. So that is something probably needed to be considered. It should be fully automated, miniaturized systems, which can be also microfluidic based because they work very well in the microgravity. So the crux is that there is increased need of automation because the limited crew time that is available on board, either ISS. Thank you. So what we need to do is we need to bring the lab to the sample. So point of care testing is the solution. A lab in a chip is the solution. This is microfluidic based. Lab in a chip technology is microfluidic based. In clinical chemistry, there's something known as closing the brain-to-brain loop. That is from the clinician to the laboratorian. So it is important what test is required and how is the test interpreted. So that can be done with the help of tele-laboratory medicine. And there are some technologies that we use is AI-enabled clinical decision support system, which can also help in the onboard flights. So there is specialized training needed for space lab medicine specialists because this is a very small branch, I would say, of lab medicine, and we do not have many experts in this area. So they need to have the understanding of space medicine and essentially space physiology. There needs to be direct demonstrations because they will not be all trained in space laboratory medicine. There might be engineers working in these areas and have been conducting these experiments. This study needs to be supplemented with self-directed learning, e-learning, AR, VR aids, and there needs to be a competency assessment, which is also required in our laboratories. 6-monthly competency assessment is a mandatory thing. Documentation should be of the quality control and the QA processes. How do we ensure that we can initiate the education during the undergraduate medical education? There can be electives and clerkships in space laboratory medicine, residency and rotation also can be there. And special conference sessions on this can be organized. On both simulation exercises is very essential because microgravity experimentation in microgravity experimentation on Earth is very different. A cell culture experiment might take 10 minutes here on Earth, which will take 30 minutes on board. So that is very, very important. And case studies, how to deal with those troubleshootings on board. That is very, very important. So thank you all for the attention. This is a podcast which was supplement to the paper that we had. So if you have any questions, I'll be happy to take them.
Thank you so much, Saswati. That was a great presentation. We've got 2 more speakers. The next one is Christian Randieri from eCampus University. If you wouldn't mind, please turning on your audio and video and sharing with us.
Yes, okay. Let me share my screen. Okay, perfect. First, I am Christian Randieri. I want to say thanks to the organization for this opportunity to let us show our research in this symposium. Today, I'm going to talk about this study that essentially is leveraging natural language processing to analyze experiment cross-relation study using GeneLab and IMPA repository. Let me move this. Okay, perfect. Okay, it works. Okay. Essentially, this study proposes a novel approach utilizing natural language processing techniques to extract and analyze experimental data from GeneLab and IMPA repositories using an ad hoc developed tool called BioGene. Essentially, the objective to this study is to uncover hidden patterns and associations between experiments, supplementing traditional analysis methods, and also to try to understand complex biological phenomena through AI. The BioGene tools that we developed consist essentially of 2 modules. The first is the BioGene Hub that collect and keep updating text data obtained from NASA GeneLab, the import repository. And the second is the BioGene AI, as composed of NLP models applied to the experiment sets, by the way, data sets collected by the BioGene Hub. Now let's talk with the first module, the BioGene Hub. BioGene Hub is a tool dedicated and with dedicated and independent service that run independently to collect continuously and automatically data experiments from NASA GeneLab and import repository. This platform is built scalable in order to get involved with future integration, for example, the ESA repository. Also, adopt relation SQL and no-relation NoSQL databases for the data storage. Is composed of a friendly user interface to interact with the data stored in the databases, while the tiles and manage external documents such as scientific publication. BioGene Hub is composed of 2-phase workflow. The first phase is the context extraction phase. Essentially, the source platform API is integrated for extracting experiment text from each platform. And the second one is the data elaboration phase. Essentially, create a SQL data structure, also extract the publication text from the experiments, and lastly, generate the dataset readily for NLP processing in standard formats such as JSON, JSON-L, and others that we can implement in the future, essentially. So the content extraction phase is, as I told before, is adopt the API calls to the each repository in order to try to catch the text from each experiment associated in a NoSQL database. Why we choose the NoSQL database? Essentially because the data contained in this platform are highly unstructured, and also the APIs are of different generation. And this will assure us the maximum flexibility to future platform evolution with different API. So you can see in the screen on the left the first interface of the BioGene tool, where essentially you can see all the experiments that were extracted from both repositories, and you can see that is indicated the platform type, the experiment name, the title, and also the URL and the number of publication that the tool was able to extract. For example, in the first one, you can see was able to extract only 4 of 27 publication. In the second screen down in the left, you can see a particular experiment screen, and essentially is a summary of the experiment in terms of summary, protocol, samples, files, ontologies, and publication. Okay, let's go to the data exploration phase. Okay, this is composed of the first part. It is to create a SQL data structure. Essentially, an SQL data structure is continuously updated starting from the NoSQL database through a specific service. And this will generate all the list of the experiments that were imported. The second is the publication extraction feature. It's composed essentially of 2 methods. One is the automatic extraction that is valid only for open access papers with the working links. And the second is by manual upload. Essentially lets the users upload all the relevant publication manually in PDF formats. We will see later that only about the 15% of publication were automatically passed from both platforms. In the screen, you can see just the interface that show for the experiment OSD 6022 which kind of publication were extracted and which can be uploaded manually. Okay. Let's go to the phase regarding the generation dataset ready for NLP processing. It's essentially composed by multiple selection or single selection of 1 or more experiments. It is possible to generate the related JSON files that are ready to be processed by as dataset by NLP algorithms. In the second part, we will say this will be done by the BioGene AI. So essentially, we have a list of study. We can select 1 or more study of all the study, and then click to export all. Then the tool will produce a JSON file that is ready to be implemented in the BioGene module or in other module that people can develop. This is the statistics regarding the passed experiments. We was able to pass all the studies for both platform, all the files name, and regarding the publication, we was able to pass only the 21% of publication in GenLab and the 13% in import. This second statistics shows the overall study scrapped, that is almost about 1,500. The files accessed was about 90,000 files, and the publication passed was only 245, and others 1,398 were not passed due to the fact that the publication was not open. Okay, let's go to the BioGene AI module. This module is essentially a tool for extract knowledge from experiments, data, text, and publication. Is composed essentially of a fine-tuning process implemented over NLP algorithms. Actually, we adopted the BERT, ChatGPT, and Llama algorithms, but also the tool is scalable to other algorithms for future integration, such as Google Gemma and so on. So Just for brevity, we show just a fine-tuning process example where essentially we keep two studies in consideration, one from the NASA Gene Lab, the OSD 285, and the second is the import SDY 2393. Essentially, we put this study together and we give them to the basic NLP algorithms in order to ask them to carry out a question list. Secondly, this question list associated to the topics, by the way, the combination of the text of both were submitted for a fine-tuning process, and then we got the answers. So essentially, the answers generation is composed from the step 2. The step 2, we submitted this question to 3 kind of fine-tuning algorithms submitted to each experiment, first separately, by the way, the first time the OSD of GeneLab, and the second of the import, and lastly, by the combination both. So essentially, the question that we selected was 4, that is represented in this slide, and we gave them to the each algorithm for carry out the answers. The results was essentially that the combination of both study can improve the answer score of all the algorithms. So it seems that these methods function. By the way, by the combination of similar experiment in both platforms can improve the answer responses. Just for clarity, this is an example of the Q1 questions that essentially was what is the specific dose rate of cesium-137 using the acute exposure of cells, and how does it compare to the dose rate of the cobalt 57 using the chronic low dose radical exposures in terms of their respective physical and biological effects on the cells. And essentially, you can see the comparison of all the 4 algorithms. We reach a score of, for example, in SBERT 93%, about BERT 78, and so on. So, in conclusion, what we can say, we can demonstrate that all the text and the answer obtained combining the experiments can improve the answer accuracy, but also it's very important that all of this procedure need a human validation. By the way, actually, we did just let to the algorithm to choose the question. So also the fine-tuning implemented over the NLP algorithms must be extended to more questions that should be always verified by humans. This just for in the training phase. On the other hand, we will integrate the BioGene AI interface system in the BioGene tool with the ensemble methods in order to detect the best answers from different NLP algorithms in order to try to select the best algorithms in function of the question that give much more accuracy. Lastly, I want to say a special thanks to the BioGene research group that is composed of my students that I appreciate a lot for their efforts and the time that put in this research. So thank you for your attention. If you have any questions, you are welcome. Thank you very much.
Thank you so much, Christian. Great talk. And our last speaker here to round off the symposium is Jessica Braun from Brock University. So if you would not mind turning on your audio and video and sharing, please.
Yep, perfect. Is that okay? Can you see that okay? Okay,
yep.
Awesome. Okay, perfect. Thank you very much for the introduction. I'm really excited to be here today and kind of round off the symposium. So thank you all for staying as well. So today I'm going to be presenting, take a little shift from some of our previous talks and talk about the use of animal models to understand the cellular mechanisms underlying spaceflight-induced changes to skeletal muscle. So very briefly,
There we go. I'll just go over some background information, hopefully get everybody kind of on the same page, go through some methods and results of some studies that I've recently completed, and tie everything together and talk about some future directions as well. So when our astronauts return to Earth following several months to years in space, they demonstrate significant muscle weakness and atrophy. So I had to include a photo of our beloved Canadian astronaut here, Commander Chris Hadfield. And upon his return, you can see pretty clearly a frailty about him. And this is very common. Typically, they're not able to stand and have difficulty adjusting back to gravity. Now, in order to counteract these losses, astronauts typically maintain a very strict and arduous exercise training regimen, but unfortunately, it has not proven to be enough to attenuate the changes to skeletal muscle. So this leaves us with the need to discover an adjuvant therapy, but unfortunately, the cellular changes that occur to skeletal muscle are very poorly understood, and this is mainly due to there being such limited access to samples, especially those from our human astronauts. So our best alternative to spaceflight samples are those from Earth-based models, more commonly bed rest studies, as well as animal models, which I'll get into. So one of these studies that was recently published collected characteristic and functional muscle data from participants, and they actually demonstrated that weakness appears to precede the muscle atrophy. So specifically, they found an approximate 3 to 6% decline in muscle cross-sectional area here, whereas functional differences declined between 14 to 23%. So no biochemical analyses were performed to really dig into the mechanisms behind this, but it is hypothesized that calcium dysregulation may be involved. So briefly, calcium is extremely important in skeletal muscle function and performance. So when we have an action potential that reaches our muscle fibers, it triggers release of calcium from its storage site, the sarcoplasmic reticulum, or SR, where it's able to bind to our contractile filaments and allow for force production. Now, in order to elicit muscle relaxation, the calcium must be removed from the cytosol, and this is actively done by Circa pumps. So Circa pumps are vital for eliciting muscle relaxation, as well as maintaining low intracellular calcium levels. And I'm sure a lot of people are familiar, prolonged elevated calcium concentrations can become quite damaging to the cell, and in muscle could lead to weakness and atrophy. So with our interest being in calcium regulation in muscle in our lab, we wanted to investigate whether Circa dysfunction could be involved in the weakness and atrophy seen following spaceflight. So in order to do this, we were able to receive soleus muscles from the biospecimen sharing program for rodent research missions 1 and 9. Now, the soleus is mainly a postural muscle and thus is very greatly affected in a microgravity or unloading environment, as it's no longer needed to hold us up anymore. So in these missions, we had 3 groups. So we had 2 control groups and the flight group. The flight group was aboard the ISS for approximately 35 to 37 days. Now, in looking at our soleus muscle weights from both missions, we unsurprisingly see that the flight groups had significantly smaller soleus muscle weights compared to the 2 control groups. So using our calcium uptake assay, we are able to assess Circa-mediated calcium uptake over time. So that is, again, bringing the calcium from the cytosol of the cell back into that storage site. And doing this in the soleus muscle of these 2 missions, we see quite a significant impairment in Circa-mediated calcium uptake, which can clearly be seen by the red line in the 2 graphs, as well as our area under the curve measurements being significantly higher in the flight groups compared to the 2 control groups. And that is, again, indicative of less calcium taken up over time. Now, Circa pumps are very highly susceptible to reactive oxygen and nitrogen species, or ROS, and thus we wanted to investigate total ROS content in our samples. So we used the markers nitrocysteine and nitrotyrosine, and we found in both missions quite clear elevations in these protein markers, demonstrating increases in ROS. So together, and based on our available data from these 2 missions, we concluded a working hypothesis whereby during spaceflight, exposure to radiation and an excess of oxygen due to muscle unloading is leading to an excess in ROS production, which might be causing damage to the circa pumps, impairing calcium uptake. Now, this damage actually induces a bit of a cycle of destruction, whereby reductions in calcium uptake by circa can actually further drive ROS production through increases in cytosolic calcium. And it just creates this vicious cycle of damage there. Whether this is leading to or contributing to the muscle atrophy and weakness that we see following spaceflight does still remain unknown. But this led us to another question along the lines of whether preventing or reducing ROS could potentially protect circa function and improve muscle health following spaceflight. So in order to answer this question, we were actually able to obtain more soleus muscles, but this time from the Rodent Research 18 mission. So this mission similarly had 3 conditions, so 2 control groups and 1 flight, but these mice were further divided into recipients of either saline treatment or BUOE, which is an antioxidant. Now, importantly, a recently published paper using retinal tissue from these mice did indeed demonstrate that BUOE treatment was able to reduce 4-H&E content, which is a marker of oxidative stress. Now, in looking at soleus muscle weights, however, we only found a main effect of spaceflight reducing muscle weight with no beneficial effect of BUOE. Regardless of that, we still investigated circa function. This time we were looking at circa ATP hydrolysis ability rather than the calcium uptake assay, simply due to some limitations with sample preparation. We didn't see any effects of spaceflight or treatment on maximal circa ATPase activity. But what we did see was a significant reduction in the ionophore ratio with spaceflight. This ionophore ratio, without getting too detailed, is essentially a surrogate measure of membrane calcium leak. So we can actually get an idea of how much calcium is leaking out of its storage site into the cytosol of the cell. So again, a reduction in this ratio signifies that spaceflight is increasing calcium leak from its storage site, which would then be increasing cytosolic calcium concentrations. So for consistency with previous work using the retinal tissue from these mice, we also investigated 4-H&E content, again, a marker of oxidative stress, and we found a main effect of flight increasing 4-H&E, and we actually didn't see an effect of BUOE in these samples. So to look a little bit further into this, we also investigated protein content of SOD, which is a natural antioxidant enzyme found in our cells, and BUOE is actually a mimetic for this protein. We did find reductions in SOD content following spaceflight, again, no effective treatment. So we're hypothesizing that perhaps the reduction of SOD with spaceflight is offsetting any sort of potential effect of BUOE on reducing 4-H&E and ROS content. So with this data, we concluded that spaceflight increases ROS production, which not only damages circa pumps themselves, but also increases calcium leak into the cytosol. Unfortunately, and contrary to our hypotheses, BUOE was unable to attenuate these changes in skeletal muscle. So putting together the data from the 3 missions that we were able to work on, we are concluding that circa appears to be consistently impaired following spaceflight, and this may be due to increased ROS production. Unfortunately, antioxidant treatment with BUOE was unable to attenuate the markers of ROS and circa dysfunction in the soleus muscle. Now, before I conclude, I just wanted to bring up the idea of open science and how great this symposium was. I think the idea of open science greatly contributed to our ability to investigate these cellular changes that occur and also be able to compare and share data with others working on similar or even same missions. I think that this really drives collaboration, innovation, and is, of course, accelerating scientific progress. And so, again, I'm very grateful and excited to have had the opportunity to speak today. And again, thank you all for sticking around for the last talk. So before I wrap up, I just wanted to acknowledge and thank my lab members, the Free Outer research team, as well as everyone involved in running these missions and our funding agencies. Thank you.
Thank you so much, Jessica. That was a great talk. So we are at our Q&A portion for the section. So if I can ask all of our speakers to please turn on your cameras. I will go through the questions in Conference.io as they are directed to the specific speakers. So first speaker here is Andres. Would you mind coming on camera, please?
Sure, here I am.
All right. So are there treatments on Earth that use mitochondria transfusions? If so, could these be repurposed for spaceflight?
Yes, there are several initiatives that are trying to understand the effects of the best source of mitochondria and to fuse them in circulation. So there are several mice assays, for example, those that are taken by a joint venture between a company, which is Matrix Bio, and universities on Paris that are working on this part. Yeah, they seem to have good effects on the hippocampus of aging mice.
Wonderful. Next question is directed to Saswati.
Hi.
Great. So your question to you is, will microgravity impact the shelf life of drugs? And will it affect the bioavailability and eventually the efficacy of the drugs?
Yes, there have been drugs that have been studied there, and it will impact and it will depend on the drug that is being tested. There is a ISS formulary which is available, and also it gives there's a list of what is the bioavailability of the drug, of the drugs that have been used in the ISS formulary, that is there. And there are a few experimental drugs. There are space pharma experiments that are being done. So those are isolated data that is available, but not all of the drugs that we use on Earth have been studied there, but it will impact.
Wonderful, thank you. Our next question is for Anu, but Anu, this particular question you already answered in the chat. So the question is, please explain the clustering step again in more detail. For those of you that are interested in that question, can I please refer you to the chat? Because I think Anu has a lot of other questions. Want to make sure we get to them. So the next question, Anu, for you is, what is the interpretation of correlation loss during space flight, and how does it relate to health and disease states? Anu, are you on?
Whoops, sorry, I was muted. Yeah, that's a great question. The way we interpreted the correlation loss that we see during spaceflight is that it's almost like a loss in regulation of the genome, right? Because correlation loss is not necessarily expression loss or change in expression, but a change in expression is almost always change in correlation. So yeah, so correlation is almost like a way to measure co-expression, and so the way we to reiterate, the way we looked at and the way we interpreted the correlation loss is that it's essentially a change in the regulatory systems of the genome, and so that's why we think it's a good biomarker of the negative effects that astronauts face.
Wonderful, thank you. The next question is for the Purdue team. So the question is, since you see different signals at different organelles, would single cell data help follow up on these findings since you can look at different cell types?
Yeah, we agree. This is something that we're planning to do actually with Dr. Richard Parker. And yeah, because like the main thing is like checking different differential expression, like grouping them across organelles, then you have a little bit of an idea of how that looks like in your system, but you don't know exactly if these cells like in themselves correlate with one another, or how similar these cells are like overall. So then maybe the expression depends on like some cell type specific response. So we want to just check on that. So yeah, no, it's something we're looking to do.
Awesome, great. Next question is directed to Jessica. The question for you is, these different missions used different sex, duration, and strains. Any variation between tissues that you can infer to either of these factors?
Yeah, that's a good question, and we've thought about it a lot. So yeah, the RR9 mission was male mice, RR1 was females, and then I believe RR18 was back to males. There isn't a lot of work done regarding circa function in general across biological sex. I'm actually currently working on that right now, so I can't infer too much right now. The RR1 mission as well, we only had an N of 4 per group, which made it quite difficult to come to any big conclusions. So yeah, as for strain and biological sex, it's hard to say right now. I would imagine there would be an effect. Females and males experience muscle atrophy differently, so that is something to keep in mind. But unfortunately, again, based on that limited availability of samples, we just had to work with what we had right now.
Wonderful, thank you. We got a lot more questions to go over. So I'm going to start with Anu again. One question here is something you already answered in the chat. I'm just going to repeat the question verbally and direct everybody to the chat. But the question is, what are the biological implications of the microRNA and protein, miRNA and protein clustering that you observed? So that question is in the chat, so if I could please direct you to read it in there, as there are a few other questions for Anu in Conference.io. So the next question for you, Anu, is to address the batch effect, which normalization do you use particularly?
Yep, I can answer that. So basically what we did was download the raw data sets from GeneLab, and we pre-processed them using Llama for microarray experiments, and then FastP and StarSolo for RNA-seq studies, which I think is in line with the GeneLab processing workflow. Once we pre-processed the data sets, the actual normalization method was basically sorting the genes in every count table by raw expression value, and then dividing those count tables into 200 groups of equal size, which then transformed the count tables into rankings within each study. And then what we did was use, I think, the least square function in SciPy to identify scaling factors for each gene. And we did this by fitting each gene's expression value to the, I think it's called the root quadratic equation, where it's y equals ax squared plus bx plus c. And in this equation, y would be the expression value, and the x would be the rank of the gene in that profile. And then once you have the coefficients for that equation, you can then calculate the scaling weight for each gene, just simply using the scaling weight equals 2ax plus b, where you know, a and b are the scaling factors that you identified earlier, and then you just multiply that scaling weight by the expression value to obtain a normalized expression value that you can then basically combine amongst the gene transcript ID. I do see a question in the chat asking if the RNA sequencing datasets were single cell. I do not know off the top of my head, but I don't think so. The StarSolo package that we used did seem to work well for the datasets that we had. But I can follow up with you on that, Amanda. And again, just to provide some more information, I can also put the chat, put a link to the preprint in the chat if you have any other further questions for the methodology.
Awesome, thank you. So our next question here is for the Purdue team again. The question is, are there any ways to combat these changes in photorespiration when growing plants in space?
Yeah, so that's the motivation of the study. Like, that's what we're trying to find. Like, that's a really interesting point. That was what we were trying to, like, hoping to find, kind of like characterizing if it was an adaptive or reactive response. because plants have shown to grow in space. So we want to understand like the mechanism of how they do it. And actually, by pure serendipity, we were mapping a pathway for carbon fixation, I think it was, and we did not realize it was actually for a C4 plant, which is C4 plants are adapted for this like reduced photorespiration. And the genes that appeared in the C4 specific pathway were actually differentially expressed in some of these studies. So we don't fully know exactly what's going on, but we know that there might be a hint of that the plant is somehow adapting to this environment. So that's what we're looking to find.
Awesome, thank you. Next question is for Christian. The question for you is, oh, it just moved. What are the potential implications or applications of the findings from this study for researchers and practitioners working in fields related to genomics, immunology, or data analysis?
Okay. Yes, this is a great question. Essentially, this is a new idea because to try to merge different databases from different experiments, for example, GeneLab and IMPA is very challenging for research because I believe that different study can be compared. One can be done in the heart and the second can be done in the space. So it's interesting to try to understand how this kind of study can have a matching point in order to enhance the data interpretation for students, for researchers, and so on, and also to try to accelerate the involved research. And lastly, I believe that these tools can be a nice tools like an AI search engine, for example, because we can try to ask a question to the system and the system can try to address us to the related experiments that may be correlated where people can find different solution questions and data to work on. So essentially it's this. Thank you.
Great. We have one more question from Andres here. The question is, did you look into nucleosome positioning like Chris Mason and Daniela Bezdam did?
Thank you very much for the question. No, we haven't, but we look forward to it.
Awesome. And our last question here is for Saswati. Question is, what are the most promising technologies that are currently being developed for space flight medical procedures?
So if it's medical procedures, it's not just lab technology we are talking about. So the most promising is the ultrasound that we have, ultrasound capability that we have as of now. And also I know there is a group that is studying, they are trying to develop MRI, which can be done. It's not yet tested on microgravity, but that is something that is to be seen. Portable X-rays that might be available, that can digital X-rays that can transmit the data. And if you talk about the laboratory technologies, Oxford nanopore sequencing was tried on ISS. That was something really good. Sequencing availability on board ISS, that is something excellent. And also mini PCR was tried in ISS. And there are lots of groups studying tissues in space and actually studying the whole genome, trying to do the whole genome sequencing in space. So these are some of the very excellent technologies that are coming up. Organoid studies in space, that there is a project. Tricia Leroux, one of our colleagues, she is leading that project. So these are some of the things. There are many more. I guess I'm forgetting to mention maybe some. So do reach out if you need any of the resources. Thank you.
Thank you so much. And that it concludes our Q&A portion for this section of speakers. I just wanted to thank all the speakers for your time and your wonderful presentations today. And I'm going to turn it over to Salon Cost for some closing remarks.
Okay, thank you everyone It was a great second day. I hope you really enjoy it as much as I did. I tried to take notes, and I'm really impressed by the amount of work that comes out of open science. And I don't know if some of you are musicians, but I was always surprised by music because you have this very limited number of notes, and you think all melodies have been played, and suddenly you see people taking the same notes and trying and coming up with new tune, new song. It feels the same with you guys. I've been looking at this data quite a bit with our team, and yet you are able to take it, play with it, and turn it around and come up with new ideas. And I'm looking forward to the next phase, which is uh, converting this into true knowledge. And I think it will take the shape of some kind of like logical diagram at some point that will be able to really understand many of these features together. You can see the puzzles coming along, mitochondria. So we heard a lot about this in the past few days, but I really feel like it's getting much more complex, and it would be really great at some point to start plugging in some kind of like logical diagram describing many of these features into a cohesive view for us to teach the space biology to others. So that's something that's floating in my mind right now as I was listening to all of you. So thank you everyone to make the AWG and the symposium so live. You are this project. This is living through you. So we can't thank you enough for all the hard work you do, and don't hesitate to reach out to us if you need help, if you need support. We are trying our best. Paola said it very well. There's a lot of investment from NASA to maintain open science, so people don't realize this, but there is really quite some effort to make it happen, to keep the database running. And so we're really trying our best to deliver to you guys as well. So I hope you appreciate the work we do as well. So with this, I think we can close this symposium, and thank you, Lovorka and the entire team, to put this together. I know it's a lot of work. That's another thing that people may not realize. It's the coordination, setting up the meeting, reading the abstract, selecting. All this stuff takes time and effort. So thank you, Lovorka and Marco and everybody else, and the great team at OSDR. So hopefully I'm not forgetting anyone, Lovorka or Lauren, whoever is online that wants to add to what I said. But thanks.
Thank you so much. And I greatly want to thank the OSDR team, Lauren and Danielle and Sam. They really helped make this symposium come alive. So thank you to all the speakers for taking the time to speak, and for the public for listening. Thank you all, and I hope you had a great time.